All right. Why don't we go ahead and get started? As I said, thank you everyone for joining us today for the Knowledge Portal Network webinar. Today I'm going to go over some news items and then I'll hand it over to Trong, who is our featured speaker. So with that, our first event that we'd like to highlight is the Anvil Community Conference 2026, which is going to be held August 31st through September 1st at the Broad Institute. Registration is now open for the community conference. And if you are interested, please feel free to scan this QR code, or you can email me directly so I can send you the link if this is not the easiest way for you to apply. And then one thing I do want to note is if you are planning on participating and you would like to submit an abstract, please do so by the 26th. I know that's coming up in a few days, so I just wanted to highlight that now. And if you're interested in learning a little bit more and you want to see, some of the details, please feel free to scan this QR code, here, where you can learn a little bit more about the event and the speakers. So if you're interested, please feel free to join us. Next, I just want to highlight that our Pink-based office hours will be coming up on August 20th and October 22nd. The topics are to be announced, but if you're already on this Zoom, don't worry, you won't need to register since it'll be available using the same link. We will share details on this as soon as we have them, but we want to have you put this on your calendar while you have the opportunity to do so. Our next webinar for the Common Fund Data Ecosystem Public Webinar Series is going to be held on Friday, July 31st. And we will be having the Human Reference Atlas, or HRA, talk about their 3D multi-scale biomolecular human reference atlas construction, visualization, and their usage. And the speakers are listed here. So we'll have Caddy, Andreas, and Daniel join us for this session. So please feel free to register for this event as well if you're interested. And so with that, today's presentation is going to be on rare variant association studies, a primer, a problem, and proposal for baseline masks from my colleague Trang at the Broad Institute. So with that, I will hand it over to Trang.
Thank you, Mackenzie. Let me try and share my screen. Can you see it
Yes, thank
in the
you.
slideshow mode?
Yes.
Okay, perfect. Thank you. Hi, everyone. My name is Trang, and I'm a computational biologist at the Knowledge Portal Network. And I've been working with Mackenzie and Julie and Noel and Jason for 5 years now. And recently, one of our research projects was published on Nature Genetics on rare variant association studies. And today I want to share some of the research in that project and happy to discuss and take any questions as well. So feel free to just interrupt me by putting in the chat or the
Yeah, we
messages
have we
that
have
you
the
want.
chat
Okay.
Q&A, and then if you want to ask your question via the Zoom, and you can raise your hand and we can
Yeah,
allow you
got
to
it.
talk as well. So yes,
Yep.
we'll be monitoring that throughout.
Perfect. Thank you. All right. So going over the agenda a little bit. So like Mackenzie just introduced, there's three parts to this presentation. The first one is the primer. So I want to go over what rare variant association studies are, and a couple of important concepts such as burden tests and masks, which are the main topic of today's talk. And then I'm going to go over a problem that we've discovered as we were working on some of the RVAS before, which will be in the literature review section. And then finally, I'm going to talk about the proposal that we have to increase the consistency and reproducibility in RVAS by going over a lot of different masks or masking strategies that we tested in our studies. And then finally, I'm going to go over some, if we have time, I'm going to go over some other considerations beyond masks and masking strategies in burden tests, and finally just reflect on what we've learned from the studies. All right. So, first of all, I'm going to make a broad assumption that many of us are familiar with genome-wide association studies or GWAS. And GWAS are typically equated to common variant association studies because most GWAS filter on variants with minor allele frequency of larger than 1%. Um, on the contrary, we also have exome-wide association studies or XWAS, which are typically equated to rare variant association studies, because in these studies, we typically look into variants with minor frequency of less than 1%. Um, and most of them are coding variants. So I'm going to use those concepts kind of interchangeably throughout the talk. Just a very quick side-by-side comparison between GWAS and XWAS or CVAS and RVAS. These are complementary approaches to figure out which regions of the genome, which variants, which genes are associated with a certain trait or a disease. So whereas in GWAS, we often focus on common and non-coding variants, in exome studies, we do associations for rare encoding variants, typically. In GWAS, because these variants are usually common, a lot of them get inherited together, so they're more likely to be affected by LD, which is linkage disequilibrium, whereas in RVAS, it's less of an issue. When it comes to pinpointing effector gene, because in a GWAS locus, if you see a significant signal, a lot of genes could be in that region, and the variants are in the non-coding region. So it's very hard to really pinpoint which genes are actually being affected by these variants that you see the signal for. On the other hand, for XWAS, because the variants we're looking into are coding, they are usually in the coding region of a protein of a gene, and it's therefore the gene is directly implicated. Another advantage that exome studies have over GWAS is that we can infer the directional effect between a gene and a trait. However, they're usually less powerful than GWAS, just because historically it's been easier to sequence the genome with the non-coding variants, and we have GWAS for a lot more traits and diseases compared to exome studies or XWAS. And finally, the level of association tests for GWAS are usually on the single variant. So you get one set of summary statistics like beta, effect size, and error between each variant that's tested and the trait. Whereas for XWAS or AVAS, you not only can do the single tests, but people typically aggregate these variants, which I'm going to talk about later, onto the gene level or sometimes region level to increase the sample size. So this is just an example to demonstrate the advantage that RVAS has over GWAS. So in this GIG-YF1 locus for type 2 diabetes, we have some genome-wide significant signals for hemoglobin A1C right here. Not for T2D itself, but for hemoglobin A1C. But this locus is quite complicated. So without any prior knowledge, if you're just coming in blind and see this locus and you see that many variants are associated with HbA1c, but it's really hard to really know which is the effector gene in this locus. So it could be GIG-YF1, could be GNB2, POP7, EPO. It's really hard to know. But with RVAS, because now you have the information on the coding variants, you can test the association between those coding variants and the trait. But again, because RVAS is not powerful enough, because these variants are quite rare, you just don't have enough sample size to get any significance. For example, right here, if you just test the single variant association between the coding variants and T2D, there's no signal. And there's a one variant that's barely significant. So one solution that is very common in rare variant association studies is that we group the coding variants within that gene together, and then you test the association between the gene and the trait itself. So now you get one set of some stats. So you have the p-value, in some cases, beta effect size and odds ratio, etc., between this gene and this trait. So now you know, like for certainty, that this gene right here, GIGAF1, all the coding variants in it are collectively associated with type 2 diabetes. And then you can do the same thing for like the other genes in, the other genes in this locus. And if you don't see any um, association between the coding variants in those other genes, then it's likely that GIGYF1 is the affected gene and not the other genes in the locus. So that's one of the ways in which RVAS has been extremely successful in supporting GWAS signals and helping us figure out the effector genes in a locus. So then that begs the question of, so how do you choose the variants to include in the gene level tests, right? There's a lot of variants out there. Many of them, actually most of them, have no phenotypic impact. If you choose, let's say, all all synonymous variants within that gene, you're probably not going to see any signal, because by definition, the synonymous variants don't really have any impact on the phenotype. So then you have to really choose the right variants to include. And this is extremely important, especially in burden tests. So in the paper, we focus exclusively on burden test, which assumes that the variants that are being grouped together within the same gene have the same effect, or at least the same directional effect, before you test the gene and the trait. And the reason for this is, in the test, instead of having the variable, which is just the value for the variant, you now have the value for the entire gene. And if the variants that you're grouping together have opposite effect, then it might cancel each other out. So even if this gene is associated with this trait, part of the gene might affect the traits differently. And if you happen to include the variants that affect the traits in opposite directions, then the effects cancel each other out and you end up seeing no signal at all. So a strong assumption of burden test is that you kind of should aggregate the quote unquote right variants together. But that is a challenge, right? Because in reality, we don't really know for sure the impact of all variants that are ever sequenced. And because of that, in the field, we have developed a lot of different bioinformatic prediction algorithms to predict the impact of these variants. So for example, VEP is a very popular tool that researchers have used to get the predicted effect of each variant. And then within VEP, there's many other software or tools that you can use as a plugin. For example, DBNSFP is a library that has the variant impact predicted by a wide range of algorithms. So all of these in the diagram right here is just the main or the popular bioinformatic algorithms that people have used to predict variant function. So then we define, because there's so many different algorithms, we have come up with different ways to combine all of these algorithms, right? Because let's say you have SIFT as one algorithm and SIFT predicts this variant as damaging, but another algorithm might not predict that as damaging. So which one do you trust, right? So usually people have combined a lot of these algorithms together to get what is called a mask, which is the main topic for today's talk. So a mask is just a filter. You have a lot of variants. A mask is just how you choose which variants to aggregate in your test. So mask is a combination of filters on both the bioinformatic annotations as well as the minor allele frequencies of the variants. So one example of a masks is a loss of function rare mask. So in this mask, you only want to, for example, pick and aggregate the variants that are predicted to be frame shift, stop gain, essential splice, etc. And they have to be with a minor allele frequency of less than 1%. So only 1% of the people in this particular study have these variants. So then a masking strategy is defined, we define it in our study as a combination of masks. So on RVAS, people typically use more than just one mask because they want to test the association between the genes and the traits, but basically you're like slicing different parts of the gene. So maybe in one part, you only want to focus on loss of function, and then in the other aspect of the gene, you want to focus on only the damaging missense variant. So people like to use a couple of masks in the same study and then do a bunch of aggregation tests afterwards. So just to recap, the primer, RVAS are useful counterparts to GWAS because it can pinpoint the direct genes associated with the trait, and it can also give the directional effect between the gene and the trait with burden test. And then in RVAS, we typically aggregate rare variants on the gene level to boost power. And to do that, we first have to define masks, which are the filters on the variants. There's a question? Yeah.
Yes, there's a question in the chat. Any existing tools to predict gain of function?
Uh, Not that I know of. It's I think it's very hard to predict that. I do know that there's a couple of database out there, for example, MaveDB, that might have some gain of function prediction, but that's limited to like very few genes. I think so. But yes, in web, I think the main annotation is still loss of function and damaging or not for missense variants.
Great, thank you.
Thank you. Right. So any other questions for the primer before I move on to the second P, which is the problem? Okay, perfect. So then the problem that we observed kind of anecdotally in the past was that lots of different studies use lots of different masks, and these masks were not completely different from each other, but they're also not completely the same as each other. So for example, there's 3 landmark UK biobank studies that have association tests for exomes and a wide range of traits by 3 different research groups, and they all use different masks and masking strategies. And when we saw that, we just wanted to see, okay, so does it really matter, right? Like, even if they use different masks, do the results actually differ from each other? So we just kind of got the published associations from the browsers of these 3 studies for the 400K to 500K samples. So the samples are roughly similar to each other across the studies. And across 30 plus something traits that we look at, there was only 28% of result overlap. So that means that after correcting for the number of masks in each study, and then you get the significant outcomes across all of these traits, there's very moderate overlap between, which genes are considered as significantly associated with which traits across these 3 landmark studies. So that motivated us to really broaden our question and our search to really extensively kind of determine whether, this is how big of a problem this is in the literature. And we know that it was a problem, as in like we knew that people use different masks all the time, but we didn't see any other publication out there that kind of actually um, examine this in a systematic manner. So we decided to did a literature review across hundreds of studies that conducted a rare variant association test, and we examined hundreds of masks across all of these studies, and we also documented them and we harmonized them, and we did all of that to make sure that we could now use them in our test and test the compare the end results, which is the associations between the genes and the traits. Um, so we found that all of these masks varied a lot in the definition, right? So like what is considered a loss of function variant? That varies by a lot. What is considered a damaging missense variant? That also varies a lot. They also vary a lot in the tools that the researchers use to annotate the variants, the allele frequency reference database, and because of all of these, differences in masks, to simplify and to harmonize all of these, we decided to like adapt all of these definitions within one single pipeline, so that we still kept the definitions of the masks, right? So like what is considered a loss of function, but um, we only apply it within our one single annotation pipeline to make sure it's consistent. So this is just a brief overview of what kind of masks out there. We noticed that there was a couple of themes or categories that emerged. So again, loss of function historically has been used by a lot, which is the blue area right here. And then people also use a lot of damaging missense masks, loss of function and damaging missense in the same masks. Only missense and indel variants, and some studies included basically all coding variants altogether. And then the second type of mask that we noticed you could also stratify these according to minor allele frequency or MAF. There's many different ways to kind of bin these MAF into different bins, but we like to, in this study, we define the common variants as with MAF of greater than 1%, and then low frequency is between 0.1% and 1%, and then the rare is MAF less than 0.1%. So again, low frequency variants historically have been used by the most, and then followed by the rare variants. And then the masks that use common variants typically belong to like the older studies before we could sequence like hundreds of thousands of people. So after we kind of collected all of these masks, we tried to figure out the overlap between the mask definitions across the studies, right? Because we want to see whether or not in reality people, did reuse the masks from other studies and try to reproduce the results, or though they just made up like new masks. We found that what we thought was just a one-off observation turned out to be an actual widespread problem, because we found that this is a bar plot. The y-axis is the proportion of studies that use the mask. And then, the y-axis is the proportion of the masks or masking strategies. And then the x-axis is the number of times that mask or that masking strategies was reused. So most of the mask and masking strategies, like more than 80% of the masks and masking strategies were only used once. It means that for the majority of the masks and masking strategies, nobody else outside of that particular study would reuse that. And not only they did not try to reuse existing masks, they also did not justify their masking choices. So basically, you don't have a reference point to start. You create your own masks, and you don't really provide any like reasonable justification as why you choose this mask or why you choose this combination of algorithms and not the others. And many studies actually referenced previous studies. So it's kind of a vicious cycle, right? Like there's no guideline, so there's no reference point. So you just end up either choose the previous studies which did not justify their choice, and then or you come up with a new mask and then also not justify your choice of mask. Um, so then we look into one studies that was followed by other studies a lot. And in that study, in the supplementary information, they clearly stated that in the absence of a clear gold standard, we took two approaches to combine the metrics. Um, so this is a study from 2014, which is more than 10 years ago, and it's been 10 years. We have so many more algorithms, and we still don't have a gold standard. Um, so that's what we found in the review. Then again, we know whether this is a problem or not. We don't know yet. Maybe it's fine to have different masks. Maybe it's fine to have different ways of grouping variants across the studies. And actually, one reviewer suggested that it might be good to have that kind of flexibility because then researchers can have more freedom in, sorry, I'm not paraphrasing the reviewer's word right, but basically um, rigid prescription is not always good. And we actually agree with that. However, there's a need for a baseline, a starting point, right? So we decided to take all of these masks that we've revealed, ran our burden tests for 54 traits in the UK Biobank, and then compared the different results across all of these masks and masking strategies. Is that a new question? Okay.
Nope.
No, okay.
Oh wait, actually there is one in the Q&A. Sorry about that. What is the purpose or benefit of masking?
Yes, so that's a good question. The benefits of masking is that let me go back to the previous study. Sorry, previous slide. Sorry, one second. When you basically, did I lose this? Oh, there you go. If you don't mask, so if you just conduct the association test on the single variant level, right? So each variant is tested with the gene, then like you normally would with the GWAS, then there's not enough power to detect any association most of the time because these variants are so rare. Um, like some variants only have 5 or 10 people carrying them. Um, so the way to increase the power is you aggregate and collapse all the variants together to increase sample size and increase power. Um, but the result of your test would depend on which variants get to be included in the test, right? So if for the same gene, if you, let's say there's 100 variants in the gene, if you include 10 variants, you would have a different result from including 5 variants or 6 variants, right? And then the type of variants that you include also affect your results. So masks are just a way to kind of declare, okay, which variants I want to group in my test for this gene. Did that answer your question?
While we wait to see if that was the answer, there is a question in both there's a chat and in the Q&A. So I'll start with the chat first since I saw this come up. Um, Hi Trang, nice chat. One dump question. If you test so many masks, what threshold should you set for the p-value?
Yes, so that is a good question. We use the thresholds. So we still use 2.5 e to the minus 6 as the exome-wide significance threshold. So you divide 105 by 20,000 genes or so, right? You get that threshold. But then in each study, you also have to correct for the number of masks in each of the studies. So let's say if I'm looking at study 1 and I'm doing the 5 tests that this study did, um, then we have to like adjust the bond failure correction, right? From 2.5 e to minus 6 divided by so like lower it to bond failure correct for that. And then you uh, decide which variant, sorry, which genes in that particular study are significant. And people usually in a lot of softwares, there's usually a function to come up with an aggregate p-value. So within that aggregation test, there's already correction for multiple hypothesis testing. And people usually do that. For example, Cauchy, there's the Cauchy test, so that whenever you run burden tests across masks in one study, it already kind of outputs one single aggregate p-value across all the masks, and it already corrected for the multiple tests. But in our study, we want to be conservative. So instead of using the Cauchy test, we just kind of bond for and corrected for the number of masks. It's a little conservative, but we make sure to be consistent across all of the studies that we reviewed so that we can have consistent results.
I think that answers one of the other questions, who had said, wouldn't this be overly conservative? So I think we
Yes.
got there.
Yes, in reality, yes, you probably in one-off studies, right? When you have a specific trait, you probably want to be a little bit more liberal with your correction. Yeah.
One last question,
Yep.
before you go on to the next segment. Do you think mass design should be disease dependent? Cancer versus non-cancer phenotype would be different.
Uh, yes. That's also something I found in my literature review is that a lot of studies actually did design their masks based on their disease. So, for example, a lot of studies they for cancer or a rare disease, they kind of excluded known variants so that they don't inflate their statistics for a couple certain genes. So in those cases, I do think it's actually very justifiable. So the authors in those studies do justify why they pick certain masks and why they exclude certain variants, and I think that's a great way to do it. So yes, the answer is yes. If you can justify it, then you should. Yeah.
Great, thank you.
Yep. But sorry, to follow with that, in our literature review, because we cannot, it it's it could be a lot to try to 100% replicate everything that everyone has ever done. We decided to stick with the masks that were used for like more generic purposes. So like when they don't provide any specific exclusion criteria, or we stick to the masks that we can easily replicate with our analysis pipeline. So that's why even though we reviewed like more than 600 masks, we had only a set of 271 masks in our follow-up analysis. Yeah. Perfect. Okay, I think I'm a little bit short on time, so I'm gonna try to quickly go over this. So yes, we after we did curate all the masks from the literature, we wanted to see if the very little overlap that we saw at the beginning across these three studies changed at all after we've applied the same analysis pipeline and the same samples. And we did see that the overlap increased modestly, so from 28% to 36%. So that's actually good news that when you control for all the other variables, right, your replication rate should increase, but at the same time, the increase is not too much. So it kind of still emphasizes that most of the differences in the results are probably still coming from the differences in masks and mask the set of masks that people use. So then we did the same thing. So right, like so for each study, we got their masks, we applied the tests, and we compared the associations, and we found that across the 54 traits that we analyzed in the UK Biobank, the number of associations varied a lot between studies. So if you take all of the associations, it's between 58 and more than 2,000 associations. If you just look at the associations coming from only rare variants, then it's between 58 and 352 associations. Yeah. So that is something that we've empirically validated, right? That in the literature, there's no consistency in mask usage. And because of that, the findings are also not very highly replicated or reproduced across studies. So because of that, we wanted to come up with a baseline set of masks that we hopefully could encourage people to use in future studies if they don't know where else to start. So like if they are looking for a reference point, or if they want to focus their studies on validating and interpreting the results instead of spending a ton of time and effort into designing masks with good justification. So we wanted to, come up with that set of baseline masks for people to start following. We tried a couple of different methods, but in the end, we kind of settled, not settled, but like we compared different methods, and then in the end, we decided to go with this greedy algorithm that we've developed to basically maximize the number of associations that we could discover within one study design. And then we also made a couple of adjustments to make sure that the strategy is not too big. So in the if you look at the my cursor right here, the maximal number of associations you could find from in this particular data set is like coming from a 22 mask or 35 mask strategy. That's way too many masks for people to usually run burden tests on. So we kind of lower the threshold a little bit, and we chose the 8 masks strategies as our baseline that could still provide the high discovery power, but also with a reasonable number of masks to run burden tests on. And this is a bar plot to show the number of associations in our data set, that each set of masking strategies that we've tried. So the final 2 bars are the proposed baseline masks, which are our conclusion. And then the dashed lines are the average number of associations. Yellow is for low frequency associations and low frequency mask, and blue is for rare. So as you can see, the 2 baseline masks that we're recommending produced on average twice more significant associations after correcting for multiple tests than a typical previous study. So that's already a win in discovery power. And we also looked into the individual traits, and we see that the 2-fold increase was pretty consistent across the traits that we looked into. And these are the summary table. You could get the actual definition in the paper, but basically most of the masks in the baseline strategies are either loss of function or loss of function and damaging missense variants. And the definitions are, again, like they're all over the place, right? Because we're picking these from the literature, and it's to further highlight it that there's no consistency. But at least now we know that empirically these set of 8 masks kind of have pretty robust and high discovery power. Right. So now we have we have 2 sets of baseline masks for low frequency and rare variant associations. But then how do we know that the additional signals, right? The 2-fold increase in discovery power, how do we know that the signals that we found additionally are not just noise? So we did a couple of tests to validate that. The first one is we conducted some gene set enrichment analysis to confirm that most of the enriched gene sets for these traits are actually sensible, like, or plausible biologically, and we did see that. So if the additional signals, like if the majority of the additional signals that we see are noise, then you shouldn't expect to see a lot of sensible gene sets being enriched for these traits. And then we also looked into the actual additional associations for the from these baseline masks, and we did see that many of them have some GWAS signal, which is a good thing, and then many of them also have like experimental evidence backing them up, or other source of evidence to suggest that the association we see is plausible. So for example, right here, there's an association between PTPN11 and hemoglobin A1C. It has some GWAS signal in this locus. And when we looked into the individual masks, all of them are actually in consistent direction. And in the literature, there's also information suggesting that this signal is likely real. So, for example, like the protein that PTPN11 encodes for regulates insulin signaling, and the gain of function mutations in PTPN11 cause Noonan syndrome, in which patients are prone to insulin resistance, which is consistent with the negative effect that you're seeing here in the burden tests, because the effect that we're seeing is coming from damaging variants. So that's consistent with what we see in the literature. And we also looked into other studies, other data sets outside of the UK Biobank. And in general, we also saw that the baseline masks produced more associations than the other studies that we looked into. So we looked into all of us and AMT2D genes, which are the two non-UK Biobank cohorts. We also compared the baseline results with the results from the other studies for binary traits, so for diseases endpoints. And we picked 11 diseases in the UK Biobank and all of us across the case and control spectrum. So they have, they both range from, they all range from very common diseases to rare diseases. And we also saw that in general, the baseline strategies generated more significant associations than the other three studies. Another variable to test for is also, diseases or traits across the polygenicity spectrum. So some traits are maybe caused by only 3 or 4 genes, like very few genes, and some traits are caused by many more genes, right? So we also look into that, and once again, like when we bin these diseases and these traits into different bins of polygenicity, we also saw that consistently the baseline strategies produced more associations for both rare and low frequency associations. And if you have any questions about this, I can elaborate on that about how we define these bins of diseases and traits. So we basically tested to see whether the additional associations from the baseline masks are plausible, and they were most of the time. And we also looked into their performance robustness across different contexts, and we saw that their high discovery power was preserved across different data sets outside of the UK Biobank, across different types of traits, so both continuous and binary traits, as well as across different traits of across the polygenicity spectrum as well. So then that's kind of summarize the baseline masks, right? But in the future studies, when people don't know where to start, yes, they can use the baseline masks as a starting point. But at the same time, we can also use the baseline masks for investigating other variables in burden tests. So they kind of serve as an anchor that you can use, and then you can explore other important factors in burden tests as well, because obviously there's transcript choices, which transcript do you choose to collapse? There's also which burden test software people should use in their studies. So we use the baseline masks as the shared variable across all of this to answer these different questions in burden tests in general. So there's many different things that we tested, but I want to focus on just the transcript choices. So this plot just summarizes that a gene could have multiple transcripts, and a variant could have multiple predicted effect. depending on what transcript it belongs to, right? So when you do a burden test between a gene and a trait, which ones do you choose? Like if you choose transcript A, then maybe a variant that is classified as damaging missense would go into the test. But if you choose transcript B, then it's no longer classified as damaging missense and it would not go into the test. So which one do you choose, right? In the past, there's a couple of common approaches. So the first one is you just choose the most severe consequence across all the transcript. So if a variant is classified as a frame shift, which is loss of function, in one transcript, where you can just ignore the other predictions, and then you only look at the most severe consequence and decide whether or not the variant falls into the masks. The second one is you can use the canonical transcript. The third one is that you define a set of criteria to prioritize certain transcripts over another. And in our study, we chose transcripts that are there's a criteria in web that say that prioritize the transcripts that are most experimentally validated. So we chose that one. And then the final option is you can always test all transcripts. So basically, instead of testing 20,000 genes, now you're testing 20,000 times however many transcript that the gene has. I have a question. Yes. So main transcript in our study, we did not use main in our particular study, but yes, you can when you annotate the variants, you can totally use main transcript as one of the priority criteria. Because this is not mask related anymore. This is after you define your mask, right? Then you can come up with a set of criteria to, let's say, if this transcript is supported in main, then you choose that one instead of whatever yeah. Yep. So, right. So we tested, we compared the results of the same baseline masks, right? So we applied the same baseline masks across these different transcript choices. And we saw that the most experimentally supported transcripts are kind of the best ones out there because they balance between specificity, discovery power, and efficiency. And by that, I mean, if you look at the figure on the right here, the Venn diagram, if you choose all of the transcript, so the light green panel, sorry, if you choose, right, then you kind of get almost all of the associations possibly discovered. But you have to like test many more than just 20,000 genes. So that's a lot of computational cost that you might not want to expand. If you only test the most severe consequence variants, then you get the highest number of associations because you're basically picking and choosing, right? Only the most severe ones across all the transcript. But if you do that, then your results are not transcript specific anymore. And in a lot of diseases, you only see association when it's specific to a transcript. Because in reality, the gene, before it becomes a protein, it first has to go through different transcripts. So yes, the most severe gives you the highest number, but it's not transcript specific. If you test all of them, you do get transcript specific results, but you have to expend a lot of cost computationally. If you choose the canonical transcript, which is pretty straightforward, in a lot of tools, they have a column that denotes whether a transcript is canonical or not. You get significantly fewer associations. And in our study, we chose the TSL prioritized transcript, and we saw that they recovered most of the associations, but at the same time, still pretty efficient, and you still get the transcript specific results. So that is the recommendation that we have in the paper as well in terms of transcript choices. Um, we also considered other factors in burden test. So burden test software, we found that there's near perfect concordance, which is to be expected between the most commonly used software, Sage and Regini. But Sage Gene is slightly better for extreme case and control imbalance traits because it's specifically designed to handle cases like that. We also looked into the different aggregation levels. So whether you want to collapse the variance across the entire gene length or just the conserved domains. And we saw that the conserved domains are shorter than the entire gene, so they were less powerful. But overall, there's still a lot of concordance between the two options in the results. We also looked into other aspects of masks beyond just increasing power. So we also looked into, simplicity, the mask simplicity versus interpretability. And finally, we also looked into the different frequency thresholds. I'm not going to go too deep into this, but these are like the main factors in burden tests that we also examined after we've established our baseline masks. So yeah, that's kind of the second section of the project where we looked into the different variables beyond masks in a burden test. So the conclusions that we had was that there was no guideline for rare variant aggregation methods, and because of that, the results were not very highly reproducible across studies. And this problem is probably underestimated in our study because we could not possibly really, examine like all the thousands of masks out there that's been conducted. So the problem is probably more widespread than what we discovered here. We then tried a couple of different methods and came up with a set of masks that we propose as the baseline that are high in discovery power and consistent across various contexts. And we recommended this as the starting point for researchers in ARVAS. It is not a forced prescription, but if you use it when appropriate, for example, if you don't know where else to start, you want to just do a burden test and see the associations without having to justify for that, then it is a good starting point. Or when you want to look into other aspects of burden test, like we did in this paper, you can also use them as the anchor for all of those variables as well. Another use case is if you want to create your own masks, then it could also serve as a benchmark. Like you can use the baseline mask as a benchmark to, compare with your new masks and justify the use of your new mask from there. So yeah, depending on your research questions.
There's another question in the
Mhm.
Q&A, which asks, since you mentioned that burden tests cannot account for different direction of effects, why did you use this for your paper? Did you try other tests like SCAT, etc.?
Yes, great question. We chose to only focus on, actually, I don't need to show my slide. We chose to only focus on burden tests because there's one advantage that burden tests have over SCAT and SCATO is the directional effect. In SCATO, you can only get a p-value because they aggregate the variant statistics after you've already done the test on the variance. And because of that, SCAT and SCATO are better or like are more powerful when the variants are in opposite directions of effect. But with burden test, when you aggregate the variance like that, then you can actually get a better like an effect size, which is super important in interpreting what this gene is doing to the trait, right? Without the effect size, you don't know whether it's protective or increasing the risk of the disease. So that is one major advantage that the burden tests have. And because of that assumption that the variance should be in the same direction effect, or even with having the same effect size even, masks are even more important. So masks are less of a problem in scatter scatter. Yeah.
Thanks, Trung.
Yep. Hope that answered the question. Um, right. I think that's about it. I think actually, so you can browse all of the gene level associations results that we generated from the baseline masks in the browser on the portal, and we also provide a script that you can use to create the group files for your studies as well, based on the baseline masks. And all of this information is also provided in the paper itself. So, yep. Before we end, I'd like to thank all of my colleagues, both at the Broad Institute and Amsterdam UMC, for helping me really bring this project over the finish line. Thank you. And thank you everyone for attending and asking questions. I'm here to answer any more questions that you have.
Yeah, we do have a couple more minutes for any questions. So please feel free to use the Q&A or chat or raise your hand. And if you have any questions that might be a little bit more in depth, you can reach out to us at our help email as well, which I can put in the chat.
Or just my email is fine as well,
Oh, that
Mackenzie.
works too.
Yeah.
Okay, so I'll put in Trung's. Okay. So you can email Trung specifically for that. And yeah, we'll give it another minute just to see if there are any other questions and such. I think we have had a really lively discussion already, so do really appreciate everyone taking the time to ask the questions that have been covered already. But while we wait for some other questions, I do want to remind everyone about our upcoming webinar in 2 weeks. We are going to be having Professor Chen Li from DKNet presenting, and he will be discussing DKNet Computational Core, a cloud native collaborative AI ML platform for biomedical research. And so at this session, you can learn a little bit more about the DKNet AI project that they are working on. So if you're interested, you don't have to register again. If you're already on the Zoom, you are registered. So this is an upcoming session. So with that, I will see. Are there any other questions for Trung before we close out for today? All right. Well, thank you so much for presenting on your masks and paper. Really grateful to have you here as part of our team.
Thank you. Thanks everyone for staying and asking questions. See you all later.
Thank you.
Okay, thank
Bye.
you.